and to talk to us about his work, Dr. Rubén López Valés, who's a professor at the biology department, cell biology, physiology and immunology department of the University of Barcelona. He's part of the Institute of Neuroscience, the Cibernet. He's got a PhD in biology from the Autonomous University of Barcelona and a PhD in neuroscience in 2005. He spent 3 years in Canada doing his postdoc and returned in 2009. Also interesting to mention, he was the first ALS grant of the Caixa and Francisco Luzón Foundation in 2018. He studies the contribution of inflammation to neurodegenerative processes, and specifically he'll talk about the influence of inflammation in ALS. Hello, good afternoon. I want to thank the organizers, the Ramón Areces Foundation and the Luzón Foundation, for inviting me to present the work that we are doing in the lab very briefly, and I want to thank all of you for being here and for listening. We've just heard a talk with a bit of an English accent. I'm going to give a talk with a Catalan accent, but I'm sure you'll be able to follow. And fortunately, you've already been told about ALS, which is the topic today. And one of the things I wanted to emphasize is that most patients have an unknown etiology, is what we call sporadic cases. But there's a relatively small percentage of patients, about 5%, where there's a genetic cause. About 30 genes whose mutations give rise to different forms of ALS. And to study a disease whose cause is unknown is really difficult. And the fact of finding these genes, even though they're present in only a minority of patients, have enabled us to come up with genetically modified mice with these mutations so that we could look into the physiopathology of the disease. And an important fact had to do with the SOD gene, and this mouse is not actually obeying me. This gene here was the first that was described. In fact, thanks to this discovery, the first murine ALS model was obtained. And you might think, well, if ALS is a motor neuron disease and part of this is caused by a mutation, if we could correct that mutation in motor neurons, we would expect those motor neurons not to degenerate, but that actually doesn't happen. When you correct that mutation in the mice, in motor neurons, the disease has the same progression. And that led us to realize that there are other cells which are not neurons, glia cells in the nervous system, play a key role in the disease. But as I said before, this model here, this G93A mouse, is the one that's mostly used in the labs. And it's used basically because it mimics all the symptoms of ALS. And that has enabled us to identify some of the mechanisms that are involved in the death of motor neurons. And one of them is the inflammatory response. And here you can see a picture of a control bone marrow with staining for microglia, which are the macrophages we have in the central nervous system, which look like this under normal conditions. And in an ALS mouse, and these are the motor neurons which die with this disease, you can see how many of these cells we have, they're a lot more abundant, these microglia. And that tells us that in the nervous system, at least of mice with ALS, there's inflammation. Is this present in patients? Well, as I said, this actually applies to only a very small percentage of patients, but it seems that it's present even in patients who have the mutation and also in several patients with sporadic ALS. You can see here in brown this activation of microglia cells. But again, the fact that we observe inflammation does not necessarily imply that it has anything to do with the course of the disease. It could be a cause. And so it's really very difficult to study because, as I said before, most patients are sporadic and we don't have any mice that develop sporadic ALS. But there is a study which I'd like to mention because it's very recent, which does provide clear evidence that inflammation is involved in the disease. And it's this study here, which attempted to find genes that were mutated or expressed differently in normal controls and ALS patients. And this led to the discovery of this gene here, the receptor beta for interleukin 18 gene, which is a receptor that, as you can see here, can bind to the cytokine, which has the same name. Luckily, it's all about interleukin 18, receptor for interleukin 18. And what happens is when it binds to this normal receptor and to this protein, which is a sort of co-receptor, it's like a security mechanism to know whether the pathway needs to be activated or not, that triggers an activation of the inflammatory response. And we saw that patients who have this mutation, it's actually a polymorphism. The difference between a mutation and a polymorphism has to do with whether it's more frequent or less frequent in the population. And it's actually relatively frequent in the population. And it was observed that the people who had this polymorphism in the gene had 5 times less probability of developing ALS. And one of the important findings is that very few patients who have this mutation and who develop ALS, which is a mutation that is believed to be protective, but the ones who have it have a life expectancy which is approximately 6.5 years longer. And so this study drew a very clear connection between the inflammatory response and the course of the disease. Having said that, of course, this is surprising because inflammation was always believed to be a protective mechanism. And in fact, now, after this COVID-19 pandemic, we've always tried to protect ourselves from the infection through inflammatory mechanisms, and that's true. But for an inflammatory response to be beneficial, you need to have 2 factors. First, it has to be desired, and second, it has to be resolved effectively. Because if it's not resolved effectively, or if we don't want inflammation and there is inflammation, that can cause different diseases. And in fact, in our system, when there's an inflammatory response, it has a very basic characteristic. Sorry, the slides are getting away from me. When you have sclerosis, spinal cord injury, or ALS, you can see that there's an inflammatory response which is not resolved correctly. In the case of ALS, you can see the inflammatory response really increases over time. And why does this inflammatory response resolve? And that was really one of the mysteries. Resolve or not resolve. And to do that, Harvard researchers about 20 years ago did an experiment which was about natural inflammation or peritonitis, and they collected pus in the remission phase, and they started to look at what that inflammatory exudate contained, and which they felt could determine the resolution of that, of that inflammatory response. And they identified a family of lipids, which is not the kind of fat we have in our belly. These are fats, but they're very small molecules, and they act as a signaling mechanism between cells. And these are called SPMs, which are specialized pro-resolving lipid mediators. And in some diseases where inflammation was not resolved, they saw that these inflammatory mediators which resolve inflammation were not expressed, were not being synthesized. There was a deficit. And this is where we started our project, which is the Talento ELA project, which began about 4 years ago. And where do these phospholipids come from? These lipids come from omega-3 acids, which alone don't do anything, but through different proteins which we have in our bodies, gradually mold that structure until they produce the different SPMs we have. Here are the 4 major families of SPMs. And to simplify, omega-3 would be like a piece of coal, which doesn't do much and it's not worth much. Well, actually now with gas prices where they are, it's worth a bit more. But under pressure, with sufficient pressure and sufficient time, it can become a diamond, which is extremely valuable. And so we decided to study and look at whether these mediators that resolve inflammation are being produced normally in patients with ALS. And so we collected plasma samples from patients in their first visit to the neurologist, so without any treatment for ALS yet, and we decided to look at the values of these mediators here. And to our surprise, or maybe not so much of a surprise because it was one of our working hypotheses, we realized that there was an absence of production of these SPMs. We did see some intermediate mediators, these ones here, which are not the diamond, but are something in between coal and the diamond. And these can be detected, but these mediators, these intermediate mediators have no function. And we also saw that in patients with ALS, these intermediate products were also present in smaller quantities. So they weren't producing SPMs, but also they have a lot less tendency to produce them. And as I said before, this is in patients with very diverse forms of disease. Some are familial, the great majority are not. But is this happening also in the ALS mice? And we did the same thing, looking at whether there was any difference with humans in the production of these mediators in different stages of the disease, and we saw exactly the same thing. These SPMs are not detected. We did detect the intermediate molecules, and the mice with ALS also have a reduced production of these intermediate metabolites. And of course, one of the things that you wonder is, well, maybe it's downregulated because patients have an omega-3 deficit. If you don't have the coal, you can't produce the diamond. And so we decided to assess omega-3 levels, and actually we found that no, they don't have an omega-3 deficit. They're just incapable of turning these lipids into these SPMs that resolve inflammation. And so no matter how many omega-3 supplements you take, it's not going to have any effect, because the problem is not with the raw material, it's in the mechanism that transforms omega-3 into these SPMs. Another thing we saw in this study, almost by chance because we looked at various metabolites, we found these ones here. And what we observe is that in ALS patients, this is drastically reduced versus controls. And when we looked at correlations and the progression of the disease, we saw very clear correlation. The lower the level at diagnosis, the course of the disease. So we have this as a potential biomarker for the disease. But if we go back to SPMs, you can see that they are not present, they're not being produced in ALS patients, So we have a lot of inflammatory mediators which activate the inflammatory response, exaggerated inflammatory response in these patients, but we don't have the mediators that resolve that inflammation. And so once it's activated, it will be increased very quickly and continuously. And so we decided and thought, well, if these lipid mediators suppress inflammation, and that's probably one of contributing to the progression of the disease, why don't we administer them exogenously? If they're unable to synthesize them, we can administer them, increase those levels, and see whether that halt the course of the disease or slow down the progression of the disease. And to do that, it was to establish a dose-response curve, and then did a large-scale experiment with ALS mice. And I'm going to show you some preliminary results we've obtained. Of course, we're still working on this. But one of the things we've observed is that the animals treated with one of these SPMs, what happens is that the progression of the disease, as you can see in green, slows down. And that happens in both males and females. So it is a potential therapy to slow down the course of the disease. And so here we have ALS and the course of the disease in mice. When you administer these lipids, which tend to resolve inflammation, what we see is that the progression slows down significantly. And well, I just want to thank the neuroplasticity group in AB, which is a very large team, as you can see here, and those who have contributed to the project, obtaining those clinical samples that we obtained through Mónica Povedano, and also our partner at the University of the Republic of Uruguay, who's a biochemist who can analyze these metabolites, and thanks to the funding from Caixa and the Luzón Foundation for this ongoing project. If you have any questions afterwards, I'll be happy to answer them.
Now it's time for Dr. Manuel Portero Otín, who is medical doctor, studied at the University of Lleida, and he is currently scientific coordinator of the metabolic physiopathology unit at that university. The previous speakers moved around, this one too. He's done postdoc courses in the US and the UK, Toulouse, Mount Sinai, New York. And he is a professor currently at the university, and he is very interested in the lipidic metabolism of ALS. So the title of his talk is Cell Stress and Lipidic Metabolism in the Physiopathology of ALS. Working towards new ways of treatment. Good evening to you all, and thank you to both foundations, Ramón Areces and the Luzón Foundation, for their kind invitation, and to Charo for introducing me. And as I must do, I have to thank the team behind me and the sponsors of all this, especially the Carlos III Health Institute, foundations like Miquel Valls, patients associations and what have you. The talk I'm going to divide into 4 areas. On the one hand, I'm going to talk about these lipids and what they are in the nervous system. Nervous system and lipids, this is very important, or we find it is important to the extent that, as you know, the fatty tissue contains the highest number of lipids, but after the fatty tissue, we have the nervous system with the largest functional and structural diversity that it has, or the larger functional diversity. And lipids in the brain contain a very high amount of polyunsaturated fatty acids, omega-3, which Rubén has talked about before. And they have a characteristic which is that in addition to be a source of resolution mediators or inflammation mediators, they're very easy to oxidize. It's what we call the oxidation of fats, which has been known for a long time. And you would think in an organ that has so many fats that can oxidize, it turns out that it's an organ that doesn't really have a lot, cannot defend itself very well from oxidation. So why might that be? We don't know. And to add complexity to all this, it's a system. It's a set of cells that consume a large amount of oxygen. This is the structural diversity of lipids. There is an endless number of molecules that are lipids in the nervous system, but not just structural diversity, but we have also diversity in terms of location. Here, this is a representation of the distribution of lipids in the membranes, and this is just the structure. Neuronal, glial, or microbial membranes are a true labyrinth, a maze of structures, and in order for them to work properly, they have to have certain structural characteristics. So how can we study this endless complexity? Well, through collaboration with experts like Dr. Isidro Ferrer, who's a neuropathologist, and using a tool that's called lipidomics, which is basically try to see what types of lipids and what number of lipids we have in the samples of interest. Thanks to this technique, we've made progress in the knowledge of the disease. And one of the things that we looked at first was is ALS a single entity from the point of view of lipidomics? That is to say, all patients that have clinical characteristics of the disease, do they exhibit the same lipidic profile pattern? We know that clinically they are very diverse, but does this also reflect at molecular level in lipidic terms? And we studied together with Mónica Povedano and Victoria, Ariona and Joaquin, we studied a series of patients and we studied the lipidoma not just in the plasma, but in the spinal fluid, and to see whether the lipids were different. And yes, it turns out that they have different lipids from healthy people. And what struck us was that it's not just that they had different lipids, but they had differences in plasma lipids. This is evidence, further evidence of what Dr. Pozo has just told us, Del Pozo, about the importance of metabolism, peripheral metabolism, in the genesis and development of the disease. Now, what lipids were they? Can they be used as biomarkers in a small series of patients? We don't know, but these lipids divided using many different routes. A huge amount of different routes, necroptosis, ferroptosis, etc. And if you look at the causes of the disease, there is a parallel between the processes that affect these lipids and the genes that are involved in biosynthesis or apoptosis, etc. But what we did was not just differentiate patients from controls, but also we wanted to see whether patients in whom the disease advanced more quickly had a different lipidic profile. And yes, the answer is that it does. through a blood test, maybe tomorrow in the future, we'll be able to help patients to know how fast their disease is going to develop. And we've detected a series of lipids that correlate very well with the concentration of plasmatic neurofilaments, which are considered as one of the indicators today of the lesion. And in addition to that, we have achieved a situation where part of these lipids, when we add them in vitro to a motor cell culture from humans, there changes in the length of the neurites change. There are changes in the length of neurites in vitro. And we think this can all be very important in terms of the different phenotypes of the disease. From a point of view, a wider point of view, the importance of omega-3 fatty acids has been mentioned. We know that omega-3 fatty acids change their composition depending on where they are in the patients of the disease, not just omega-3, but some of the modifications that are derived from them. Here we see an accumulation in the spinal cord and also the brain cortex. And also, this is correlated, here we see in samples from post-mortems with a certain increase of those resolution markers that I spoke about earlier. But also, there's an accumulation of these markers that are not so beneficial, but rather they are telling us that there is oxidation of these lipids. So what we looked at was, well, we see there can be a change in the manufacture or the level of these omega-3 fatty acids. Now, what about the machinery that produces these omega-3 fatty acids? Because normally in the textbooks, they tell us that these fatty acids come from diet, from what you eat, from the metabolism of the liver, and from there they go to the nervous system. But we saw that there are tools, molecular tools. There are enzymes, the COX-1 enzyme, for instance, that are present in these samples and that have alterations as regards TDP43. So we postulated that in some way, the system tries to produce DHA in order to produce omega-3, in order to try to improve the survival, but that might have an undesirable effect, which is lipid peroxidation. So we could have a vicious circle that feeds back into itself. And the association with omega-3 has been shown epidemiologically. There are data since 2014 that tell us that a low intake of omega-3 fatty acids can be a factor that can partially explain the risk of developing ALS, but a high intake of omega-3 fatty acids could protect you from ALS. In the experimental models that have been presented, how does this happen? Well, to solve this hypothesis, or answer this question, we took mice and gave them high or low levels of omega-3 fatty acids in their diet. And the omega-3 in the diet modified their survival, but not the way we thought. They worsen their condition in this model, especially in female mice. So there is an interaction between the female mice, the intake of omega-3 in mice, and the development of alterations in this model. And we had a series of findings, and we saw that in some way this excess of omega-3 fatty acids could alter the normal dialogue between mitochondria and the nuclei of the cells. And speaking of the nuclei of the cells, which is where we keep our DNA, should exhibit alterations during ALS. That's been described especially in the nuclear pore complex, but since we've seen alterations in the lipids, we looked at the possibility of there being alterations affecting the nuclei of the cells. And thanks to the work of Omar Ramirez and Pascual Torres, we isolated nuclei from post-mortem necropsies. These are nuclei, you might say, that remain behind. They don't show you what happens in vivo, but that's what we have at this time.
And what we see, in fact, is that the bioluminescent nuclei have a different composition than those controls. And this composition is spread out amongst multiple classes of lipids. Here we have a list of all the differential lipids in this context, and some of them tend to group around molecules that we could call plasmalogens, and these are especially abundant lipids in the central nervous system. And what we saw was that the levels of plasmalogens and the enzyme that produces them, in this case in the experimental model, were reduced. And not only that, but also that when we subjected motor neurons to oxidative stress, there was a reduced synthesis of plasmalogens, which is curious because it is known that these plasmalogens have an anti-oxidation role and role in signaling. So we described how this altered synthesis of plasmalogens, which may be a pathway to study potential therapeutic targets, is altered in ALS. But it's not just that. Speaking about lipid droplets, but in this case about lipid droplets in the nucleus of cells in the central nervous system, we saw that there are lipid droplets inside the nuclei, and it seems that they are synthesized abnormally. Since we don't know what they're doing there, we don't really know whether this is a response or it's purely an epiphenomenon. But at least it was one of the findings that showed that there was an alteration in lipid metabolism at this level. And these are the three main lipid families: ether-type lipids like plasmatogens, acylglycerides, which I haven't talked about, and triglycerides, which may be altered in the lipoma. And since we had these nuclei, we decided to study some other components of the lipidoma, which are nucleoporins, and these were altered as well. And it connected more or less with the alteration of the other lipids. And we see that this alteration in nucleoporins brings about a loss of the autophagic capabilities connected to the assembly of a structure which coats the nuclear membrane, which are the nuclear pores. And since these nuclear pores are involved in autophagy, and since we knew that TDP-43 is one of the most relevant proteins in maintaining multiple genes, what we decided to study was whether some of the proteins involved or affected in ALS, which is ATG4B, might be connected with lipid metabolism. In previous findings, what we had described was that patients with ALS have increased levels of some ATG4B messenger RNAs, and that's a very relevant gene in autophagy. And later, what we saw in some data which we're now analyzing is that the levels of this protein are altered in the disease, and it's highly present in motor neurons, as you can see here. This is a healthy motor neuron, and this is a motor neuron with these deposits. And the lower intensity of the green is compatible with lower levels of ATG4B. And that plays multiple roles in lipidation, in the formation of vesicles, and in the processing of other molecules. What this makes us think is that TDP-43 is very relevant in this process, and thanks to the work done by Ana Fernandez, we're describing how the loss of TDP-43 leads to an alteration in the cell lipidoma. And each of these circles represents a lipidic species whose concentration is altered in cells that have loss of the function of TDP-43. So the conclusions, we can see that the lipidomic profile could be relevant for a better classification, staging, and prognosis for the patients. The role of omega-3s in the diet is complex, and it could contribute to neuroprotection, as had been previously shown, but also to an alteration of the nucleus mitochondria dialogue through lipidic peroxidation, changes in the nuclear membrane, lipids and nucleoporins, where the peroxisome is also involved, since that's where most of the plasminogens that I mentioned earlier are produced, may be involved in the neurodegeneration. And TDP-43 has a new role, if it didn't have enough roles already, which is to regulate lipid metabolism. And that's all. I want to thank you, and I hope to see you all in Barcelona next year. And we will have this NCALS meeting with the participation of the Foundation. Thank you. And to finish, we're going to hear from Dr. Óscar Fernández, who's got a degree in biochemistry and a PhD in biochemistry and molecular biology. Currently, he's main researcher in the Genomic Instability Group of the National Center of Oncological Research. He's worked in cancer for a very long time, but now he's focusing very much on ALS, and that's very good news, since he obtained the award for the best European researcher under 35, and I think one of the most relevant publications has identified him as one of the most influential under 40 researchers in the world. He's now trying to identify the neurotoxicity mechanisms that affect motor neurons in ALS, and the title of his talk is Investigating Neurotoxicity Mechanisms that Contribute to the Death of Motor Neurons in ALS Patients. Okay, thank you very much for the invitation. I want to thank the Foundation for hosting this event. And I'm from Bilbao, so I can add a bit of a Basque accent, and then we'll have the whole country covered.
A ver que me aclare. Vale, nosotros cuando yo volví a España, reclutaron en el centro de oncología, aquí muy cerquita de aquí, cerca de las Cuatro Torres. Y durante 10 años trabajamos mucho en un particular tipo de estrés, oxidativo.
In ALS, we talk about oxidative stress, but in cancer, we talk about replication stress, which happens when cells replicate very quickly. And for many years, we developed many animal models for different types of cancer, preclinical models to test different treatments. We did a lot of research in molecular biology and cellular biology, and we also developed different potential drugs, some of which worked very well, which we then passed on to the pharmaceutical industry so they could develop drugs, some of which are in use. So we're very happy that in the ongoing clinical trials, some of these agents we identified are now proving to be effective. And then I had a sort of life crisis, what to do, and all this work, and I started working in a lab in the Karolinska Institute, and we were thinking what we should do next, what disease we should focus on. And at that time, I decided to start to do research on ALS. And the reason was because the foundation, like the Film Foundation and others, and the Ice Bucket Challenge, like everyone else, when I saw this, it sort of awakened my empathy and thought, oh my God, what a terrible disease. And I started reading up on it, see what was known, what wasn't, and I got the feeling that most of us who'd been doing research in cancer and had some interesting tools could contribute a slightly different perspective, which might be useful. I'm not going to explain the disease once again, but from oncology, when we looked at the research that was being done in clinical development, cancer, of course, we do have a lot of very advanced ones. Thought we could still develop many more, and of course no effective treatments. That was frustrating, and also how very little we knew about the mechanisms of why these motor neurons were dying. In oncology, we had learned why tumor cells were growing or multiplying so quickly and when they wouldn't die. In motor neurons, it wasn't known. We knew that there were mutations or that they were full of vacuoles, but we didn't know what was happening, and we thought that we could contribute something with our approaches. And another thing we found attractive is that, as you've heard, first mutation that was identified was in this gene, SOD1, which is a gene that had to do with oxidative stress, and that's why a lot of research focused on oxidative stress. But if you kept an open mind, you realized that a lot of these other mutations affected proteins that have something to do with RNA function. They bind to RNA and they have a lot of influence on its activity, and that hadn't been studied as much. Of course, then there was the discovery of C9, which was, of course, a game changer because that was a very frequent mutation in patients, and so we felt that it could be a significant factor. So there were all these proteins which we felt had something to do with RNA. And in fact, when you look at the different publications on ALS and the different diseases affecting motor neurons, in these patients, a lot of the things that are described have something to do with RNA. These patients have these cells have an accumulation of stress granules which contain RNA, and they have translation problems, and they have transcription problems, and they have splicing problems and nucleolysis. All of these are problems associated with the metabolism of nucleic acids, and specifically RNA. But how this brings about the death of the motor neurons wasn't clear. So this is the C9 mutation, which has already been discussed. Robert explained in a bit more detail the work that he's been doing, which is brilliant, how the loss of function of this molecule can bring about increased inflammatory processes. We were very interested from the molecular biology point of view in showing that this mutation in C9 eventually produces peptides, some of which contain a lot of arginine. And we thought that was really interesting, first because biology, this was very new. This is a protein that was being expressed from an intron, which is an area which should not be producing any proteins, but here it is, and we thought that was interesting. And also, I have to admit that a paper that really got me into ALS was a paper by Stephen McKnight's group, publishing that these peptides, which you can buy, which in theory are present in ALS patients, you can buy and you can reduce cell death by introducing these in the nucleus of the cells. And the fact that someone was able to publish an article with such a vague title, I thought meant that it was really important. The title really tells you that we don't really know what's happening. You can buy them, you can stain them in green, and the peptide ends up in the nuclei, which are these red areas inside the cell nucleus. And the other thing that was useful was that it killed the cells, and it didn't just kill neurons, it killed cells. The reason why it affects the motor neurons more is because of the expression of this gene in those cells. My first question was, if there's a toxic peptide, why is it toxic? What makes the cell die, and what can we do about it? These were the questions that we had initially, and so our original plan to do several things, one of which was to try and understand why this peptide was toxic, try and recover it, and develop an animal model for this peptide, help us understand why this happens, and to find maybe some treatment. The clinical phase, of course, we don't want to use issues. Person brains can really try out different things. And so first, I think we have a pretty clear idea of how the toxicity of these peptides could work, which we published last year. And these peptides contain a lot of arginine, and that's the protein that has more RNA affinity of all. And in fact, molecules that bind to RNA contain a lot of arginine, and so what we think happens is that these peptides, which are these orange pins here, toxic peptides, bind to RNA with so much affinity that it displaces all other proteins, including the best known ones like P43 or FUS or others. So it's a sort of glue which binds to the RNA and stops it from functioning properly. So another thing I'm going to tell you is that we also try to contribute to drug development, which is what I really wanted to do. This is part of the work that we're doing in Sweden. And one of the first things we did when we arrived there, there's some high throughput microscopes, which are automated microscopes, which enable you to take thousands of images automatically, and then the computer can do all the counting and everything. It's called high throughput microscopes. So what you do is you take some cells, you add this peptide kills them, and then you can screen them with any drugs and find some that will prevent the cell from dying. You count the number of cells that remain in the plate, and if you find one of these cells where there are more surviving nuclei, in principle you have found something that improves the viability of cells exposed to this toxic peptide. So we did that, and we found some peptides. If you're curious, one of the experiments in the paper, but the peptide, as you can see, each blue dot is a cell colony. If you add the toxic peptide, the cells die. You see there's almost no blue dots left, but with 3 of these drugs, there are a lot more surviving cells. So in theory, in vitro, of course, this seems to work. It's all very preliminary, it's not in patients, but it's interesting that 2 of the first compounds are PRD4 inhibitors, which is one of the things that David said they're also looking into for other reasons. And butyrate, sodium butyrate, although surrounded by a lot of controversy, is this combination of phenyl butylate, which is the last drug that has received fast track approval by the FDA. And so I think it's an interesting finding that some of the compounds that we're detecting and that could be effective at this level have been found by others in very different approaches. And the Karolinska, it's very difficult to do research with mice for legal reasons. It's really hard in Europe in general, but in Sweden it's practically impossible. But they are letting us work with fish still. And so we've done some experiments in fish, and we used these zebra fish. We knocked out or remove the corium, and then you can put the peptide in the water, and when you do that, it goes into these fish cells and the nucleolus and inhibits translation and kills the cell. And we tested our compounds with this model, and you can see that at least 2 of them have a marked effect. It's a live embryo, so if you add P22, they die, but 2 of the treatments improve survival significantly. And this is still very much a preclinical trial, but it's still interesting. And encouraged by this initial screening, which was only from a repurposing collection of already known drugs, we decided to do a bit broader screening, biggest ever in Sweden. We have 37,000 compounds in our library right now. And after a lot of effort, we've come up with 2 chemical series we called Salsa and Sals B. It's nothing to do with the dance. It's about Stop ALS Series A and Stop ALS Series B. And we hope that some of of these may contribute to treating the disease, and we're working very hard with these compounds. And finally, just to give you an idea of what we've done in animals, we were curious about knowing what would happen in an animal if we had a very high expression of this peptide. It's expressing it with a genetic trick which enables you to connect to the animal, and that induces the expression of that peptide in that animal. And when we use that trick, we see the peptide expressed in every organ. In this case, this is the 1.0 version of the strategy. We had a version in which first we expressed the peptide in the whole body, not just in motor neurons. And now we're fine-tuning it so that it's just in motor neurons. We have a zebrafish we can do this in, but in this original version, it was expressed everywhere. You can see that it's expressed everywhere in the pancreas, the intestine, the spleen. And then when we do that, the cell nucleolus, as you would see in vitro, was observed by McKnight. That's altered. There's no translation, so in an animal, we're replicating what you can see in vitro. And what happens is that there's very accelerated aging. Here you can see this as a mouse where we have induced the systemic expression of these peptides. It's aging really fast. Develop cataracts, osteoporosis, fat deposits in the bone marrow, very thin skin. So, like a very old mice, very old mouse, 20-month-old. And when we do a molecular analysis, it's connected to that previous point about hyperactivity. Molecular marker of these mice is hyperactivation. This pathway, mTOR, and this is a pathway that's connected to metabolism. Hyperactive mTOR, this mouse that's aging so fast, it's extremely fast metabolism, which aligns with these other findings. And of course, if that's what's happening, then that suggests that the first thing we need to try in order to improve the situation of these animals is to inhibit this pathway and see if that can improve the outcome. You can do this reasonably easily with this inhibitor, rapamycin, which is an endpoint. And in mice, that works really well. This is the survival of these mice. They live approximately 200 days, but if you treat them with rapamycin, they live 400. I'm being self-critical because I think rapamycin is not a molecule that will work. In flies, in worms, it's worked a lot at prolonging longevity and improving the course of some diseases, but it doesn't work in human beings because inhibiting mTOR has a good side and a bad side. If you inhibit it, it has a good side, which is that it improves survival, but it also inhibits insulin signaling and growth hormone signaling. And so the holy grail would be to try and come up with a drug that could do the good things that rapamycin does in humans while avoiding the bad things that it does. And that's something we're very focused on in Sweden and also in the Institute here, to see whether we can come up with a drug that will have the beneficial effects of rapamycin in human beings, but without any of the deleterious effects. And that's potentially what we're looking at now. And my last slide is the tool in ALS. Well, we don't know yet. We're just starting this project, but we have seen that looking at data, neurons gained from ALS patients, and we analyzed these with computer models. These are public databases. Luckily now, with all the research efforts, there's a lot of these databases where you can model these questions. And if you change, if you look at C9 motor neurons, you can see that there is this hyperactivation gene called pathway. Although it's very different strategy, at least it looks pretty simple. And that's all I wanted to share. This is the team in the Spanish Cancer Research Institute. The people with the red arrow, people who are also participating in the ALS research. Our latest recruit, also funded by La Caixa. Anna was here, and she's got really exciting data from some other strategies we're looking at to improve the course of the disease, at least in mice. And this is the team in Sweden, and in red, people who are working experiments. And I know there are patients and patient families here, some of whom I knew and others that I met here. I just want to convey this message of optimism. What you've seen here, although I understand that a lot of these things might seem very confusing, or what a lot of us have been showing these curves. That's not something we could show 10 or 15 years ago. So we're getting there. It did happen earlier in cancer. I saw with my own eyes how 20 years ago some strategies which were preclinical occurred moving. These drugs that are in use already in the clinic. So by funding research efforts, a lot of us will get it wrong and so on, but only by funding many will some be fortunate enough to see what they're doing and what they're developing really help treat the disease. So thank you very much to everyone who's making it possible.
¿Se me oye? No, creo que no. Ahora sí. No, pero ahora no. ¿No? ¿Sí? Sí. Bueno, vamos a ver ahora, a dar la oportunidad de ciertas preguntas, cuestiones que podamos discutir un poco, si es necesario. Y antes que pasemos a ese turno de preguntas, de nuevo, voy a mensaje para llevarse a casa, que tiene que ver con lo último que ha dicho Óscar.
Which is connected to what Óscar was just saying at the end of his talk.
Aquí, lo que
And that
habéis
is
visto,
the reason why we're here and the things you have heard are about a lot of different mechanisms that have place. That's crucial in basic science.
Quizás ahora lo que nos está faltando es un poco una visión integrativa.
And now perhaps what we need is a bit integrated view, more physiological view of what's going on in the crosstalk between different tissues, so that we can eventually understand what's really going on. By discovering different mechanisms, we will eventually be able to integrate things. So again, a message of hope for patients and for families. The level of knowledge we have now is unthinkable just 7 years ago. So I think that's definitely a positive note. Are there any questions?
Muchas gracias a
well, thank you very much to all the speakers for their talks.
Tengo una pregunta
I have a question
al doctor
for
Óscar Fernández
Dr. Óscar
Robert Baloh,
Fernández
en relación
for
con
Dr. Baloh
de esta enfermedad, si
about
realmente es una
selectivity
única
of
enfermedad
this disease, if it really
sobre
is a single
las motoneuronas
entity
medulares,
affecting
las células piramidales,
motor neurons and cortical pyramidal cells, and the findings that you've presented on the mutations of ORF72, you've explained a lot of potential mechanisms of action. I seem to remember 3. Dr. Baloh spoke about an immunological dysfunction of dendritic cells, which gives rise to an activation of microglia with a deposit of complement in the central nervous system and a reduction in synapse activity. Did you see any specific alterations, neuron fatigue, or any alteration in the cells that are the object, that is in the motor neurons? But also, you've explained about the production of this abnormal toxic peptide, which has something to do with the RNA and the cell nucleus and altered signaling pathways affecting all cells. But why do the motor neurons die? What's special about them? Why is it so selective at that level? And we don't see any alterations in the cerebellum or in any other parts of the central nervous system.
Well, the motor neurons and in Alzheimer's, it's similar because it's part of the brain that deals with memory and also Parkinson's. But in this case, no, we don't know. There must be a reason to explain the two types of ALS and also frontotemporal dementia and why these patients have both. What in their environment caused this? That's the question. One theory is that, of course, sport. In sports, for instance, why do footballers have more ALS? Has it got something to do with the spinal cord? We don't know. Or another theory, but these are all theories. I don't think anybody here really knows the answer to this question, but it's something that I think has to do with viruses, and this is very controversial. Viruses have the ability to affect motor neurons, like as in polio and other diseases, and also as in frontotemporal dementia, which is very similar to herpes. It's the same regions of the brain that are involved. So there's a possibility that there's a group. It's possible that there's a series of events in the environment of the patients that affect their immune system and cause this to happen. But this is just my theory, and I don't know what happens with the RAM peptides because they're very rare in the spinal cord. So I'll pass the floor to the expert on the spinal cord. Well, we don't know anything for certain, but the two reasons that I usually debate with students that we believe contribute to give certain tropism to the pathology towards the neuron. One of them is because of the C9 gene, which is expressed more in that area, so the peptide is toxic in many other areas, but there's greater pressure in that area, so it's more damaged. The other reason is a bit more mysterious. ALS is not the only disease where there are many mutations in proteins that have to do with RNA metabolism. For some reason, there's tropism because of the motor neurons and the bone marrow. There are diseases where there are mutations in the proteins affecting the neurons and the bone marrow. Why changes in RNA have a greater effect on those cells? We don't know. One of the things that is debated is that maybe these cells have a higher demand of proteins because of the neurotransmitters that they produce and the hematopoietic system cells because they grow very quickly. But that's all wishful thinking right now. But what is true is that the gene C9 is expressed more in these areas, and that might have something to do with it. Thank you to everyone for your information. I have two questions. I'm going to start with Dr. Bob Baloh. In the clinical trial that didn't produce the results that we had hoped for, I wonder whether the patients that were given a transplant were subjected or were given physical muscle movement exercise intense exercise. And the reason I ask is because the trophic relations between the glia and the motor neurons depend heavily on the activity of the motor neurons. So I don't know whether in the future that physical exercise will be included as a coadjuvant to demanding secretion of more trophic factors from the glia. That's a good question. We don't control the activities of the patients in the clinical trial. And we had recurrences. They seem to need to have a level of strength in their legs, quite a good level of strength in their legs in order to do the trial and measure the decrease of strength in their legs. But we didn't tell them what to do before the trial. It's always a difficulty in clinical trials in general because patients have different lifestyles, and exercise is very important to delay in terms of strength in all patients in general. So that's a good point. I don't know how we could control that because it's almost impossible. We can give patients specific exercises during the trial, but it's very difficult to control them in general. It's very difficult in all clinical trials, technically. Yeah, physical exercise or electric stimulation, programmed electrical stimulation. Well, that's a mad idea, but the next question is for Rubén. The peptides that resolve inflammation, these three that derive from the metabolism of the omega-3 fatty acids, are they universal for all kinds of inflammation, all types of inflammation? No. No. So we must conclude that in the same way that there are many ALSs, there are many types of inflammation. Is that correct? Yeah, obviously. It's not the same to have an inflammation in a patient who's got multiple sclerosis, for instance, as it is in a patient who has inflammation resulting from trauma or an ALS patient. The inflammation is different. The components, the molecule cascades are different, but the lipidic moderators also act through different receptors. So the modifications that happen and that they have upon inflammatory cells in each case are also different. And we've seen, because we're working on several diseases, that there are some lipidic or lipid mediators that are effective in all inflammatory diseases that we've studied, and some are not. Others are specific to certain diseases and not others. And in fact, some of them could be beneficial in some diseases, but cause harm in others. So could we think perhaps that one strategy, valid strategy, would be to study the mechanisms of resolution of inflammation, glial inflammation? Well, in fact, that is one of the things that we're doing at the lab. I haven't read the project. I wanted to ask a question for David. We know from the work that you've shared with us that there's an activation in the BAT, and also we all know how it atrophies, the muscles atrophy or the muscle atrophies, mainly due to a lack of signaling from the neuron. But are these two events correlated? Do they happen at the same time? That's a very interesting question. In fact, we're exploring some possible connections that we know there are between the muscle tissue and the BAT. And one of the things that has to do with your first question, which is common, the death of the motor neuron, is the existence of a muscle, of a neuromuscular plate or plaque. In terms of energy homeostasis and control of the energy demand, there is much less muscle in the body. There are many fewer muscles in the body. One way to understand what is happening with a process that leads to ALS is to understand how different tissues talk to each other. And BAT, we know, secretes a lot of things, but especially certain control of energy homeostasis, which at a given time or moment can be irreversible and lead to mechanisms that are irreversible. And for Manuel, I wanted to ask, because in your talk you've mentioned it and it's come up in other talks, the difference between males and females. We know that there are differences in terms of location, fat metabolism, hormones, sex hormones that are lipid-based. But in terms of neurons, are there differences between males and females in lipid metabolism? Well, in our case, in the experimental preclinical model that we assessed, the answer is yes. In fact, estrogens in vitro have neuroprotective effects in the context of the overexpression of G93A. That is to say, they allow neurons, well, not neurons, but cells that overexpress that superoxide of dismutase can survive better. The signaling effect of estrogen on the mitochondria is a well-known phenomenon because potentially those estrogens can go through the blood-brain barrier and have an impact. Epidemiologically, please correct me if there are any epidemiologists here, the incidence is slightly higher in men, and that with the menopause evens out. There is not such a great difference after menopause, after women have the menopause. So I don't know whether that means that we could give estrogens to all patients. It would be a good thing if it worked, but that might be one of the hits, so to speak, of the motor neuron disease. And if that is the case, it might be very important. And talking about epidemiology, I wanted to start with Rosario. The difficulties that we have in Spain to know how many patients there are, we need a registry. I'd like to know your opinion about this, how beneficial it could be to have a registry and how difficult it is to set up a registry. But in your point of view, what could we do and what could this registry contribute to the advance of science? Well, a neurologist might be able to answer that question better because they are more familiar with the health system. But my opinion, after talking to experts, is that it is a difficult problem unless we have human resources to deal with it. I believe maybe neurologists that do clinical work know what the situation is, but it's key to have a registry. That would be a huge advance, and to support the different hospitals so that registries can be set up. And if we had a common database, it would be wonderful. With the regions, I don't know to what extent that is possible, but it's all a question of starting. That's my opinion. Good, I'm going to make use of the opportunity. I'm a neurologist, as a matter of fact, and I work in Catalonia, and the truth is that we're trying to create a registry, at least in Catalonia. The main problem, I think, is that there is no global protocol in the whole of Spain on how to evaluate the patients, because we get patients for trials, and the way maybe to assess patients in one hospital might be different from the way that they're assessed in another hospital, different scales, different ways of assessing patients. So the first thing we need is a Spanish national protocol so that everybody does the same, and we can then use those values to assess our patients and register them. I think, for instance, in France, We recently had a meeting with a neurologist from France. They have a global national protocol. Hello, good evening. My family, 24 years ago, there was a case of ALS. My family, it was the first time that I had to face that kind of disease. My mother, it was my mother. In 2 years, she was diagnosed. It took another 2 years for her to be properly diagnosed, and then she died. And my question, I always turn this round in my mind, can we rule out hereditary factors in this disease? Could somebody develop it if some relative or a parent has had it before? Is there a hereditary component to the disease? Well, I can answer maybe the question. What is clear is that most patients don't have a family history of the disease, and therefore the likelihood that a relative of somebody who had ALS develops ALS is quite low. But I must also say that years ago, people used to die a lot. People didn't know what they died of. They said, well, he was taken ill and he died, and we didn't know what it was. But now, with the advance of genomics, we're beginning to identify new genes. What is true, and I think most people agree with this, scientists agree with this, is that the big genes that are involved in ALS and that are present in the greatest number of patients are identified. Other genes can still be identified, but they're minority genes that might be present in very isolated cases and rare cases. But what is important when you get a patient is to know whether they have any of the known mutations of genes. We sometimes do screenings for some of the genes, but not all of them. So, is there any likelihood that a relative of an ALS sufferer who develops ALS in the future? It's a low likelihood, but it's not zero. We cannot hear this speaker. We need the microphone, please. I'm the daughter of an ALS patient. My father died from a neurodegenerative disease too, and I don't know whether there's the same genetic load as there is in cancer. Óscar, I think, as your name, can tell us, because I think you've been researching into that for a long time, because in my family there's been a lot of cancer. The case of my cousins and their children, there's a lot of cancer, and the whole family has got cancer. Is there any possibility that the same genetic load exists? My father had a neurodegenerative disease, my mother too. Is it possible that I might develop some kind of similar disease? Because I'm a bit afraid to do the test, the prick test, because I'm a single daughter and I have to take care of my mother. And I was talking to Stella and to Miriam at the Luzón Foundation, who are delightful, and they really give me a lot of support. And Dr. Jarabo, I don't want to do the test because I'm afraid to, but do you think I might develop the disease? Well, if you ask me, well, I'm not the right person. I can't tell you, I can't give you medical advice because I'm not a doctor, I'm a basic researcher. What I can tell you is what's been said more than once. Mutations that penetrate more, that are more associated with hereditary ALS are well known, and it's easy if you do a screening test to know. And if you've got a mutation, that increases the likelihood. That doesn't mean to say you're going to have it or get it. But in the case of cancer, it's the same. There are some hereditary mutations in families, but not everybody in the family develops cancer. Yeah, the children of my cousins. They've done the test and they got a high likelihood of developing cancer. Now you're talking about cancer. Wait a minute. Well, there are connections. I had slides in my talk, but I took them out about this. If we talk about cancer, in the case of patients with C9 and in general in ALS, there is a lower rate of cancer than in the rest of the population. So there's an anti-correlation regarding cancer, perhaps because the immune system depending on the situation of your immune system, some people might be more susceptible to immune diseases. So there is sometimes a correlation, but it's not, severe enough to make to make people feel that they should be particularly afraid of developing the disease. But regarding your other question, in the US it's slightly different because families are all over the place. But it's common to find patients that don't have a family history of ALS, but they have a gene. And that is why we in our clinic, we consider all kinds of patients. But we need resources for further research. We need grants from central government in order to do our research. And that's what the neurologist in the audience said. We need a consistent way of measuring patients, and it's very important. In the US it's almost impossible, but in Spain I think the families are not so scattered all over the place, and it would be easier, and the population in general is not so scattered, and it would be easier. It's a different situation, but I think there are opportunities here in Spain that we don't have in the US. I wanted to ask another question regarding the census, the registry of ALS patients. I think that it would be a great idea. In the regions, it might be a bit more difficult because each region in Spain is managed in a different way, but beyond a registry, I think it would be much more important for all doctors to know really what factors lead to the onset of the disease. When there are behaviors that can be identified before they develop the disease, because I remember somebody who was operated for a hernia, disc hernia, and I know a 52-year-old man who's been operated for a hernia and has ALS, and the course of the disease has been delayed because when he was operated for the hernia, that has led to atrophy in less than 5 months, so there's been a direct influence on his disease. So I think that there's a lack of information in general, and sometimes this disease is addressed or discovered after a diagnosis has been made which is completely erroneous, and the patient hasn't been given the care that they needed because the diagnosis wasn't correct. Well, if you allow me, I'll answer two things to your comments. First of all, 4 or 5 months ago, we met at the 12 de octubre hospital in Madrid. The registry is going to be set up. Well, I said that very quickly. We're going to try to do our utmost to set up the registry. There were neurologists from all over Spain, researchers, and we thought it was key. And regarding the second thing, I don't know whether when Manuel and David have spoken, they've mentioned biomarker. Many groups in the world and my group too, are working are searching for biomarkers. Something we don't mind who finds it. If we're lucky, all of us will find 20 biomarkers. Something that allows the neurologist or the physician to look at the situation and diagnose ALS. A diagnostic biomarker and a prognostic biomarker. Because we know, if you know the disease, that in some people it moves faster and develops faster than others. And that's very important. So there are many groups of research that are looking for these biomarkers. But we can't do it overnight. But we have made progress. Neurofilaments, there are a series of things that are being developed. Well, thank you very much, and congratulations to all of you. It's been a wonderful session, and it's a wonderful evening. Thank you very much. I wanted to, regarding the registry, I wanted to talk about a couple of initiatives that might be of help. Registry is one thing, that is to know how many patients there are, or a census. It's one thing to have a census of patients and another thing to have a registry. In census, when you talk about census, the Spanish Confederation of Dementia, practically all the health councils in the country are collaborating to prepare a census of people with dementia in Spain. It takes time, it's complicated. Data are scattered, there's not a single repository, and I presume that with ALS it would be much easier because most of these patients are admitted to hospital and they're diagnosed in hospital. Another very interesting experience that was recently presented at the Spanish Multiple Sclerosis Foundation, they found these difficulties, and what they've done is they've done a search on databases, all kinds of databases. Some of them are not directly related to healthcare. For instance, people who are off work, on sick leave, and they collected data that allowed them to increase the number of people that they thought had multiple sclerosis in Spain. So these are initiatives that could be of great interest. And if the effort is made in dementia, it could also be made for other neurodegenerative diseases if we take that step. And then I have a question, a very general question, which has to do with a very peculiar or characteristics kind of ALS, which is the dementia, Parkinson, ALS complex, the WUM complex, from Guam, an old or a former Spanish colony of Guam. This complex of dementia, Parkinson's, and ALS. I don't know how many people or cases have been collected, but there must be a tremendous amount. The role of C9, lipid alterations. Could we study all this in a population? Could we look at the viral prionic hypothesis in the context of this variety? The Guam Islands are very far away, and possibly the US is nearer and can study them better. Well, one of the things that we've done, one of the theories that has to do with a higher frequency in Guam has to do with a neurotoxin. And it struck us at one stage. I was skeptical regarding neurotoxins, but when I saw the data, the correlation seemed to be very high. We tried to buy in vitro experiments done by Annabelle in the lab. We see that the properties of this neurotoxin at cell level are similar. What the neurotoxin does is similar to what we see when we do the FUS mutation. The cell undergoes the same process. We don't know how the neurotoxin does it, but it does it a similar thing, and that's all that I can tell you. We know no more. Yeah, specific BMAA toxin. It's like an amino acid. That's because it was used in a clinical trial in the US. looking at a competitor of this false amino acid, but it didn't work, that trial. So the theory was that there are levels of BMMA that can be associated with the disease, and there are complicated theories that say that this toxin was carried by flying bats, was found in bats, that the indigenous populations used to eat on after the Second World War in Guam. But after that, the rate has come down to the normal rates in the rest of the world in the Guam Islands. So there are tissues, there are theories, but of course we can't do everything we'd like to do in this respect.
Well, nowadays, the problem is that when we talk about ALS, we're actually talking about many, many different entities. I won't say almost as many as patients, but really the juvenile form, and depending whether it starts with the sarthria, then there's the familial forms, then there's the ones that are associated with environmental factors. According to Al-Chalabi's work, who's really a tremendous expert in this field, he says that there are various hits on the system, and if you have a certain genetic predisposition, then you might develop the disease. And so with all of these elements in the environment, the intake, exposure to environmental toxins, it's really very difficult to know whether you will develop the disease or not. And I would agree with my colleagues that I think we can be reasonably hopeful, because we are seeing how in some trials, in some experiments, for these SOD1 mutation cases with the antisense agents in some patients, it's working reasonably well. But of course, we still need to really identify which patients. Otherwise, using very targeted therapies might come in too late. How do you fix that? Well, we're already hearing how here registries, international cooperation, having a single sample collection protocol to identify patients and monitor them. Unfortunately, of course, all of that requires a lot of funding and infrastructure, but anyway, at least we know how we might do it. Yes, good evening. I wanted to talk about funding, because there are representatives here from 5 different research groups. We all know how well-developed molecular biology is in Spain, but thinking of what happened with the COVID-19 vaccine, is there some type of research that you would like to do, but you can't because you don't have the funding? Well, question I think I would turn around and say, what wouldn't we like to research if we had the funding? It's just that the problem we're facing is actually even bigger, because even when you have the funding, it's extraordinarily difficult to use it in the right way, because it's public funding and the mechanisms and the requirements you have to meet. And I know it's the same in many other countries, or it's even more bureaucratic in other countries. But here, absolutely everything is the responsibility of the main researcher, and that's not the case in other countries. So you can be killed off by success, because even with a lot of funding, you could be overwhelmed by all the paperwork, and it can become a complete bottleneck. And so I think both the patients and families and ourselves, scientists, should start demanding those in charge to simplify all of these requirements, because we need to focus on our research and not on all of these paperwork. We don't have enough support staff to manage all the paperwork surrounding the funding we get. So perhaps the patient associations should also help us in this campaign to lobby and improve that too. I'm sorry, I can't hear that comment. The microphone. Because generally, it's a 4-year term, and politicians don't really look beyond that. And of course, science is more productive if you have plenty of funding. If you want to build a road, but you only have funding for the one bridge, that's what you're going to build. But if on top of building the bridge, you have enough resources to finish the road somewhere else, then you can have research assistants and people pitching in ideas and thinking of other avenues that could be explored. And you can't do that if you only have just enough money for the project you're in. And unfortunately, that's how most of us are operating.
Hola, buenas tardes. Bueno, primero gracias por todo.
Good evening. Thank you for this wonderful
y
evening and everything that we've learned and heard. I am an ALS patient. I have a SOD140H mutation. And the question is about the treatment Dr. Robert was talking about and Daniel also mentioned. I started receiving it in July, and Manuel said that it seems to be working. And of course, we have great hopes. I just wanted to hear your opinion. And also, as for other patients, my ALS is genetic and expressed. In 2013, I started having difficulties walking. In 2016, 2 cousins of mine were diagnosed, were siblings. One was 43, one was 49. And I was diagnosed with ALS because Dr. Vázquez, I'm in Valencia, and I had been limping for 3 years, and I was going to have surgery for lumbar stenosis. But then the course of my illness is slow, as you can see, but my family, we had never had anybody with a neurodegenerative disease. Nobody. I mean, my grandparents died of old age. And yet we've had 3 cases in my generation. One of my cousins died in 2018, his brother died in 2019, and I'm still here. But just those who were wondering whether you could develop it or not, it is pretty erratic. And the course of the disease is also pretty unpredictable, very differently in the 3 of us who were cousins. Very close cousins, and so it really is tremendously variable. In our case, Doctor, they're studying, luckily for researchers, my mother had 10 siblings and they're studying the whole family. And hopefully that can help in some way. But you can, or you may or may not develop it, and you may be a carrier. My mom is 80 and she's fine, and she's the carrier supposedly. And her eyesight's not great, but otherwise she's great. And yet, in my generation, 3 of us have developed it. I also wanted to hear your opinion on the treatment that I'm receiving. Variability in the family or the effects of the treatment? It's 2 different questions. It's pretty common, but this is one expression. There's some mutations that have tremendous variability. And it can affect you when you're 90 or when you're 20, but there are others which are very consistent. and typically more severe. But of course, the question is that it all affects neurofilaments, that's clear. As for the clinical progression, the results are a bit less clear. But probably that's why there's that variability. There's more variability with SOD1 than we thought. And they had to monitor the patients for much longer. The first 6 months, they didn't really see any effect, but after, I think it was 12, they saw an effect, but it wasn't statistically significant. But probably, after a lot longer, it will be more significant. But I don't know what the others think. But we believe that we need to do more with this gene, because the reduction was, I think, about 50% in cerebrospinal fluid. And so we probably need to do a bit more. We don't know why it's a gene which is so highly or upregulated, but it's probably that, but that's all I can say. Well, maybe just to say that we have the impression that ALS is as genetic or as ungenetic as any other disease. What I mean is that the genetic diseases that don't have your typical Mendelian track, well, any disease could be genetic or not genetic to some extent. And so what matters is really knowing what the mechanisms are that generate this convergence and trigger the disease. Another thing is that when we started the treatment, because in theory it would have been better, of course, before the disease started, and that's what they're trying to do now. But as you know, that requires a lumbar puncture every month, which is pretty difficult. But of course, it'd be best if you have biomarkers, which would tell you when to start. And so they're looking in the blood every 6 months or so when neurofilaments are elevated, and once they are elevated, they start the treatment. But is that enough? We also need to treat the inflammation, because even with this, we'll probably need to do other things as well. Well, as you said, if you're 3 cousins, you're the one who has had the best course. There's also variability in drug response. Some people do better, some people do worse. It's hard to predict. But let's hope that you'll be a good responder. It's sort of like a poker game, where you're handed some cards, and then you have to play them. But it's pretty unusual for you to get a winning hand in the first round. But some people are really lucky, and they will never get anything. They'll live to be 100 and never have a problem. And there are others who get cards which, mean they won't win and they'll develop some disease. But most of them will get pretty standard, run-of-the-mill cards. And depending on how you play the game, how you live your life day to day, you will win or lose. And then there are people, like your mother, it seems, that even though she's got all the bad cards, still wins the game. But of course, those are rare cases. And so if there's any relative or a patient with ALS who doesn't have any mutation that has been identified, most probably the fact that, or the probability of your relatives having ALS is really low. Of course, it's different if there is a mutation that has been identified in your relative. Yes, good afternoon. Good evening. It's me. The microphone might be a bit loud. First, I want to thank all the speakers for their talks. It's all been extremely interesting. And I have two questions for Dr. Baloh about the clinical trial which you showed. Have you considered any other type of strategy, not just to use cell implants, but some other vector in order to administer TDF in a way that will not be quite so invasive? Well, it is really difficult because it's a protein. If you inject it in the blood, it's not going to cross the blood-brain barrier. And so they have tried other things, gene therapy with virus, code the DNF, but that also needs to be injected with a lumbar puncture. We might eventually, and we're trying to come up with viruses that could be injected into the blood, but who would cross the blood-brain barrier in the area, in the target area, so towards the motor neurons. But so far we're unable to do that, and so we do need to do the lumbar puncture, which is very invasive. And well, in the future, I hope, of course, that we will be able to avoid that. And as for the signaling pathway, STING, have you analyzed the spleens of the mice, looking not just at the genetic and protein profile for STING, but also other mediators that might be involved in its activation? I'm sorry, I missed that question.
Yes. The gene or protein profile. Well, I understand the question. It's very interesting. What is stimulating STING in the patients and also in normal people, healthy people? We don't know. But probably in aging there are damages that the mitochondria suffers, and ADN emerges from the mitochondria, and that is probably one of the stimuli that we think is most important. And we can also detect it in other diseases, neurodegenerative diseases, especially Parkinson's. But the truth is that we don't know. And we have to measure that in mice and patients to see whether there is any correlation. Thank you. Good afternoon, evening, almost. It's very late in the evening. So the president of the Luzón Foundation has asked me to close this meeting as president, and I must, in order to do it, thank the speakers for their magnificent explanations and detailed talks, some of them above our heads, quite frankly, those of us who are not specialists in the matter, in the field. But I have to say that I've seen the bottle half full rather than half empty, or one third. One third is not much, maybe, but the bottle is getting filled more and more, and I have been at meetings for many years, and we see that the research groups are growing. There are more and more clinical trials all the time, and that research groups are more and more interconnected, and they make use of each other's discoveries. We've got Triacal, that we've got advanced drugs, and drugs in phase 3 trials, and also because there are more resources for research in the world. And in Spain, we began modestly with a program with Caixa, now, after this football game, so to speak, we're going to be able to inject quite a few million euros into research in Europe and Spain, and universities, foundations, and the regions. Here and there are interesting efforts being made, and I'll go back to one of the things that has been said. I don't know whether it was you. The line of progress or advance is reaching further and further and is increasing its speed of advance all the time. And this has to do with something that's happened in Spanish society and other parts of the world probably. For the first time, we got an association, a national group called Con ELA with ALS. It's a national association of ALS patients that has a single voice. And we are working with 3 ministries of the public administration, negotiating 23 points having to do with the needs of ALS patients. And that includes everything. And we see that there is interest to make progress. There's an advanced registry, the idea of an advanced registry, fast diagnosis system, further training for nursing personnel, specialized ALS centers here and there. And there is a strong agenda. And that strong agenda is presented, submitted to Parliament, and on the basis of consensus, there's a proposed law to deal with ALS for the first time. It's been postponed because there's another process of negotiation that is still going on, but it's going on. So the advance of research is also accompanied by an advance in social awareness in terms of the agenda that we have. Lobbying, the media have helped us a lot in the last few years. So I think that some news is good, quite good, and I hope that this meeting has helped us and helped you in your different platforms, medical, scientific platforms, doctors, patients, and groups of interest and stakeholders realize that we are making progress little by little, and this is good news. And I just want to finish by saying that the Francisco Luzón Foundation to combat ALS, we are on the warpath. We're on the warpath in terms of research, in terms of lobbying, public lobbying, with the actors and players that can make decisions, in terms of increasing research, giving further strength to associations. And for the first time in Spain, we are speaking with a single voice, and we've got Pepe, who's the national vice president of the ALS Association, and to be on the warpath means that everybody else is, as we have saw. And therefore, that's all. I have to thank you for being here, for your good spirit and for the hope that we have been given. Thank you very much.